So if there's any issues with the audio or advancing the slides, please feel free to just jump in and let me know because it's hard for me, as we all know, to get cues in this
Yeah,
virtual environment.
yeah,
So
we do.
okay, great. As Angela mentioned, I'll be talking about the Maternal-Infant Research on Environmental Chemicals Research Platform, or MIREC. So I've had the great pleasure to work with MIREC for about 10 years, starting in my PhD. But MIREC really was, I'm standing on the shoulders of giants and those perinatal epidemiologists such as Ty Arbuckle and Bill Fraser, who initiated the MIREC study. And it's really been my pleasure to continue the work that they originated back in early 2000s. So I'm going to start out by talking about the why of MIREC. Why did it get started? And this really happened, as we all know, in the framework of the development origins of health and disease framework, and the idea that what happens before birth has impacts across the lifespan. And this exposures that happen both preconception and prenatally can impact health outcomes not only during infancy, child, adolescence, and adulthood, but now we're increasingly aware of some of these effects that go into the next generation. And these effects can impact multiple organ systems, as I'll get into a little bit later, related to cardiovascular, respiratory, cognitive health, as well as impacts on the immune system. Combined with this knowledge that this early developmental window is a window of vulnerability and susceptibility to environmental chemicals is the fact that there was limited understanding of the health effects of many of these chemicals, as well as limited understanding of the levels of exposure during early life and development. So for some exposures, we have information and public health messages, such as alcohol or cigarettes. But for the vast majority of environmental chemicals that we're interested in, we really have fairly scarce data on levels of exposure and health effects. And one of those environmental chemicals that we're increasingly interested in over the last few years have been per and polyfluoroalkyl substances, otherwise known as PFAS. So I thought this would be our first poll question in terms of what are PFAS? And I see the poll has popped up. I'll wait a moment for this to register. Okay, great. It looks like that has just about stabilized, with almost 70% of people saying that they are chemicals with nonstick, water repellent, or stain repellent properties. And that's the answer I was thinking of. 40% of people said that they are plasticizers. And I can see where that answer came from, as PFAS are so ubiquitous in the environment now that they may be found in certain plastics. So that answer is actually fair as well. But as many of you clearly know, that the main properties of PFAS that are attractive to their use in household and consumer products are their fact that they're nonstick, water repellent, stain repellent. They're also heat resistant. This is, as I've mentioned, a chemical class that is of high interest to Environmental Canada. Currently, it just see if I can now go back to advancing. And you've already identified some of the sources of these PFAS. Some of the other chemicals that are of interest to MIREC include those shown in the top rows, such as bisphenols, phthalates, and pesticides, as well as heavy metals such as arsenic, cadmium, lead, and mercury that are found in drinking water, found in the air through industrial emissions, peeling paint, and certain types of food. So MIREC has measured over 200 different environmental chemicals from these chemical classes, as well as several others, such as flame retardants, PCBs. And we've measured these environmental chemicals at multiple time points in the MIREC Research Platform. So this gives you an overview of the research platform. It started back in 2008 with recruitment occurring between 2008 and 2012. Approximately 2,000 pregnant individuals were recruited during their first trimester of pregnancy. And they were followed throughout pregnancy, and they and their children have now been followed up for over a decade with follow-up studies conducted during infancy at 6 months of age, during the preschool ages. That study was focused on growth and neurodevelopment, and we're currently in the middle of our follow-up study focused on puberty and metabolic health, as well as maternal postpartum metabolic health. And I'll come back to some details on the follow-up studies during preschool age and puberty later in the presentation. I also want to point out that one of the great aspects of the research platform is the MIREC Biobank. So at each of these follow-up studies, as well as during each trimester of pregnancy, participants were asked to provide biological specimens, which were then put into the biobank and are available for future use. The overarching research questions of MIREC are really twofold. One focused on understanding levels of exposure to environmental chemicals during these windows of vulnerability, whether they be pregnancy, early life, adolescence, or menopause, and what are the effects of environmental chemical exposures on health, whether that be prenatal and postpartum health in the mother or the children's health. And so this figure here shows the 4 overarching pillars of our research. That is a focus on life course epidemiology, exposomics with our focus on multiple environmental chemical exposures, vulnerable populations, specifically vulnerable life stages, and risk assessment, because a lot of our work, particularly in understanding levels of exposure, contributes to and informs risk assessment of these chemicals. As I mentioned, we recruited approximately 2,000 individuals from across the country between 2008 and 2011, and there were 10 different cities where recruitment occurred, which are shown here. Participants had to be over the age of 18 in their first trimester, planning on delivering at one of these local hospitals, and willing to provide a cord blood sample. This figure shows an overview, a little bit more detail of the original MIREC study during pregnancy. So you can see that over 8,000, close to almost 9,000 women were approached. Of those, we had 2,000 that consented, 1,983 that completed a first trimester visit. Of those, we had 1,959 live births. What this figure also shows is that in addition to blood and urine, which was collected at each trimester visit, we also have biological specimens from delivery and postpartum, including cord blood, meconium, and hair, as well as milk. The MIREC study population tends to be fairly high to moderate socioeconomic status, primarily of white race and ethnicity, and primarily born in Canada. As well, individuals tended to be over 30 at the age of recruitment, of normal pre-pregnancy BMI, have some university education, and were never smokers. So you can see, like many cohorts that rely on volunteer participation, it's a fairly homogeneous population, and also a fairly healthy population. So I'm going to move now to talking a bit more about one of our primary research questions, and that's understanding levels of exposure. And I've put it here, these two, from an epidemiological perspective. We have one theme of descriptive epi, which is this understanding levels of exposure, and then another theme focused on etiologic epidemiology, which I'll come to later. And I want to put in a plug for rigorous, thoughtful, descriptive epidemiology. There were two papers that were published several years ago in the American Journal of Epidemiology that I thought did a nice job of demonstrating the value of descriptive epidemiology, as well as giving a rigorous framework for conducting it. These authors, I believe, were motivated by doing this work and putting together this framework after COVID, particularly when there was a lot of quickly done science, quickly done epi work that was lacking rigorous and critical thought. And mostly when we think about descriptive epidemiology, we're thinking about distribution and determinants of outcomes or health conditions. However, in MIREC, our focus is really on understanding distribution and determinants of exposures, particularly those that relate to environmental chemical exposure. I do hear some background noise. I'm not sure if someone has their mic unmuted, but if you could mute that, that would be great. Thank you. So as I've mentioned, we've measured over 200 different chemicals in multiple time points and in multiple matrices. Not only do these chemical information contribute to Health Canada risk assessments, but this information is also valuable for our ideological work as it allows us to identify those potential variables that would be confounders in our ideological analyses. So in the last couple of years, we've published descriptive analyses of several different chemical classes, including bisphenols, herbicides, phthalates, and flame retardants. And for this work, we were looking at first trimester urinary concentrations and measured these different chemical classes in the biobanked first trimester urine samples. And so having this biobank was quite valuable in that we were able to look at chemicals that we didn't originally have funding to look at, and that we didn't actually also have analytical lab methods for during the original MIREC study. And in addition to just reporting what are the average levels of chemical concentrations in our population, the key piece of this descriptive work is to understand how chemical concentrations differ according to sociodemographic and health history characteristics, so that we can identify those individuals who may be most at risk of being disproportionately exposed to these chemicals. So here I'm showing a figure from our recent work looking at bisphenol analogs. So this includes not only BPA, but some of the newer bisphenols such as BPS, BPF, and others. And what this figure shows is that concentrations in general for this sum of 7 bisphenols tends to be higher in those participants who were younger, who didn't self-identify as having a white race or ethnicity, who tended to have a higher pre-pregnancy BMI, who had lower education, less than or equal to high school education, as well as those individuals who were smokers. We conducted similar analyses looking at PFAS concentrations. In our recent work, we have measured a suite of 40 PFAS in MIREC participants during our follow-up study. So this is 7 to 9 years postpartum, looking at PFAS concentrations. Out of those 40 PFAS, there were 17 that were detected in at least 50% of participants. And so this figure is showing those sociodemographic, showing these PFAS concentrations according to reproductive history characteristics. And consistent with other research, what we're seeing is that PFAS concentrations tend to decrease the more pregnancies an individual has, the longer an individual has breastfed, or specifically what I'm showing here is the number of children breastfed, as well as the years since last pregnancy. So actually, when it's been longer since having your last pregnancy, your PFAS concentrations tend to go up. So there's this decrease right after pregnancy and with breastfeeding as it is transferred to the fetus and the breastfeeding child. But then because PFAS are so ubiquitous in the environment, the concentrations then tend to increase as this interval between the last pregnancy also increases. We've also looked at metal concentrations in relation to these social determinants of health. These concentrations here are first trimester whole blood concentrations of cadmium and lead. And what these figures are showing is that both cadmium and lead tend to be higher in those participants who had lower household incomes, so specifically those less than 50K Canadian, and also tend to be higher in those individuals who were born outside of Canada. So again, all this work is towards understanding which subgroups of the population may be disproportionately exposed to each of these different classes of chemicals. So as I've mentioned, we measured chemicals in multiple different matrices. The metals were measured in whole blood, PFAS was measured in serum, and the bisphenols and other non-persistent chemicals are measured in urine. So this brings up the second poll question, which is related to one of the challenges of measuring chemicals in urine during pregnancy. I'll just give a moment here for everyone to register their answer. Okay, it looks like these have just about stabilized. And most of the responders have suggested that the main challenge is urine composition changes throughout pregnancy. And the answer that I was thinking of actually is the main challenge we have in measuring environmental chemicals during pregnancy is related to urinary dilution. There is sometimes contamination in urine from other chemicals, but that is not specific to urine, not specific to pregnancy. Compared to other biological matrices, urine is less invasive than, for example, blood collection, and storage is a fairly routine practice. Composition may change if an individual has impaired kidney function or develops complications such as preeclampsia or gestational diabetes, but oftentimes we're looking at a spot urine sample during the first trimester of pregnancy. And the challenge we have with dilution is that depending on an individual's hydration status, their chemical concentration could be highly affected by dilution, and it could vary quite a bit from one individual who's less hydrated than another individual who's more hydrated. So one of the factors we need to consider is standardization or adjustment for specific gravity or creatinine to try to minimize the impact of dilution on our urinary chemical concentration measurements. I'm going to switch gears a bit now and move on to talking about our etiological work. And in the original MIREC study, we were interested in understanding etiology of pregnancy complications in relation to environmental chemicals. As I mentioned, the MIREC participants tend to, in general, be fairly healthy. You can see the rates of gestational diabetes, preeclampsia are quite low in this population. 2.8% for preeclampsia, 3.1% for gestational diabetes. 50% of individuals did exceed the guidelines for recommended for gestational weight gain. And we had 5.8% and 6.8% respectively individuals who had small for gestational age and preterm birth. The two complications I'm going to go into a little bit more in regard to etiology are preeclampsia and gestational diabetes. So preeclampsia, as you likely know, is a condition that is onset after 20 weeks of gestation or during the postpartum period. It's new onset hypertension combined with proteinuria and possible end organ dysfunction to major organs such as kidneys, liver, placenta, or cognitive function. We've looked at several chemical classes in relationship to preeclampsia and hypertensive disorders of pregnancy, and one of these chemical classes was, again, PFAS. And what we observed here is that individuals with elevated PFH excess concentrations had over about a threefold increased odds of preeclampsia. So these three PFAS that are shown here are three of the most common legacy PFAS. These were measured in the first trimester concentrations, first trimester of MIREC participants. We also saw that there was a sex-specific effect in this association, where those individuals who were carrying female fetuses had a slightly higher odds ratio of preeclampsia. So the literature on PFAS in preeclampsia in hypertensive disorders in pregnancy has been growing in recent years, but it was still a fairly new research question at the time of this publication. And some of the other literature showed null findings or positive findings. It's difficult to compare because of different levels of exposure and timing of PFAS measurement. A possible mechanism for this association we're seeing with PFH excess may be the role of this chemical on remodeling spiral arteries in the placenta, and disruption of this remodeling process may lead to placental stress responses and generation of reactive oxidative stress responses. Another chemical class we looked at in relation to these outcomes was the whole blood metal concentrations. And we saw that first trimester arsenic concentrations was associated with a threefold increased risk of gestational hypertension, 26% increased risk of preeclampsia. And these coefficients, or sorry, these relative risks represent the change in a per doubling of whole blood concentrations. So the concentrations were log2 transformed to represent a doubling in exposure. The literature on arsenic and these outcomes is fairly scarce, but this finding is consistent with literature in non-pregnant populations showing that arsenic is a risk factor for hypertensive disorders and cardiovascular disease. You can see that we did not see an association with the third trimester arsenic. It's possible, although we don't exactly know, that this may be due to the methylation changes that occur throughout pregnancy, and the fact that arsenic methylation increases throughout pregnancy, and methylation tends to result in a less toxic form of arsenic. That is one speculation. In relation to lead, we saw no association in first trimester, but a positive association in third trimester in relationship to preeclampsia, with a 58% increase in risk. Lead is a known risk factor for preeclampsia. However, this study extends this body of knowledge by showing this association at the really fairly low concentrations of lead that we see in MIREC. To put this in perspective, the median lead concentrations in MIREC were about 0.6 micrograms per deciliter. This is lower than what we see in the Canadian Health Measures Survey for the same time period, and in women of reproductive age, which was about 0.9. And then to put this into further context, the reference level is 5 micrograms per deciliter in the US. So we're well below that level. I'm going to move on to talk about gestational diabetes, glucose intolerance that occurs during pregnancy, and specifically focusing on our work on the metals again. This table shows the odds ratios for each of the metals: lead, cadmium, mercury, and arsenic, measured in first trimester blood concentrations in relation to odds of gestational diabetes, impaired glucose tolerance, and the combination of the two. And you can see that those individuals who had fourth quartile concentrations of arsenic had a 3.7-fold increased odds of gestational diabetes. The magnitude of this association was attenuated to 1.9 in the combined outcome, and there was no effect seen in those with impaired glucose tolerance. So this was the blood arsenic concentrations. We also looked at arsenic species in first trimester urine samples and saw a similar dose-response relationship with the one of the arsenic species and the probability of developing gestational diabetes. What I'm showing here is that this predicted probability of developing gestational diabetes is actually fairly comparable to some of the other known risk factors for gestational diabetes, such as advanced maternal age, as well as obese pre-pregnancy BMI. In this analysis, we also saw a sex-specific effect, where in this case, women carrying male fetuses tended to have a higher odds of gestational diabetes. This work confirms what has been seen in non-pregnant populations where arsenic being related to type 2 diabetes, and it's known from experimental work that arsenic can impact pancreatic beta cell function, possibly through generation of reactive oxidative species. We've also done some work more recently looking at intersectionality of this association and how this association differs in regard to race and ethnicity, as well as country of birth. Focusing on the figure on the left of race and ethnicity, you can see that the slope of this association is fairly comparable between those who self-identify as white versus those who self-identify as non-white, but the baseline risk of impaired glucose tolerance and gestational diabetes is significantly higher, about double that of the other reference group. And this persists at both low and high concentrations of blood arsenic. We see a slightly different pattern when we look at country of birth, where the baseline risk was similar at lower concentrations, but those individuals who were born outside Canada had a higher risk at the higher concentrations of arsenic, suggesting that there may be some enhanced susceptibility in this group. Okay, I'm going to move on to talk about some of our findings from our preschool, our follow-up study focused in preschool. This is known as MIREC CD+. Just to give you a very brief overview of this study, it was conducted in individuals ages, the children when they were ages 2 to 5. There was a focus of the study on biomonitoring, collecting biological samples for measurement of priority chemicals. There was a focus on neurodevelopment with both at-home assessments of neurodevelopmental indicators, as well as online assessments. And there was also maternal self-administered questionnaire, which collected information on some of those neurodevelopment indicators, as well as other factors such as breastfeeding, daycare, and maternal stress. Since the MIREC CD+ study has been completed, we've had a number of studies that have looked at environmental chemical concentrations in relation to child IQ. And what I'm showing here is a high-level overview of the sex-specific findings. And what you'll notice is that there's a consistent trend towards an inverse association between each of these chemicals and IQ, whether it's performance, full scale, or verbal IQ in males, and null associations in females. So the question that always results from this slide is, what may be an explanation for these possible sex-specific effects? So I'm going to give a few explanations focused on PFAS. So that's one of our more recent studies and one that I was more heavily involved in. And these are all potential hypotheses, obviously, that we can't prove within our data. So possible explanations relate to the toxicokinetics. It's been shown in animal studies that PFAS have longer half-lives and slower clearance in male females than females. Also been shown that PFAS accumulates at higher concentrations in the human placenta of pregnancies with male fetuses. In regard to endocrine disrupting activity, it's possible that PFAS can disrupt thyroid and gonadal hormones. And these neurotoxicants, such as PFAS, may change thyroid physiology in a sex-specific manner. Epigenetic effects are also another hypothesis for these sex-specific effects. DNA methylation may impact neurodevelopment, and PFAS has been shown to impact DNA methylation differently in males and females in at least one study. So I'm going to move on to the current follow-up study that we're doing now, MIREC Endo, which is focused on puberty, child metabolic function, and growth as our primary objective, and understanding the potential influence of early life exposure to these environmental chemicals on adolescent health. Our secondary objective is to understand long-term maternal health in relation to pregnancy complications and environmental chemical exposures. And this brings me to the last poll question, which I'll pause for a moment. Okay, and 58% of responders reported increased risk of breast cancer, which is the answer I had in mind for this question. There is there is evidence to show that early puberty also has implications for long-term cardiometabolic disease. So if you put the answer of increased levels of lipids, that is the case if we're thinking about long-term adult lipid levels and cardiometabolic disease. So I didn't make that specifically clear in my wording. So the motivation for MIREC Endo is this idea that in part that we've seen in both the lay press as well as scientific publications about timing of puberty and the fact that it may be starting earlier than it used to. And that it's been hypothesized that stress or obesity or our diet or chemicals are playing a role in this possible temporal trend, but we really don't know. This is a finding from a systematic review and meta-analysis of the changes of onset of breast development or thelarchy in recent years. And these authors reported that the age of thelarchy had decreased by about 2 to 4 years per decade over this time frame. There is some variability by geography, and no Canadian studies had been included in this, but some suggestion that for breast development, age has been getting younger. This review focused on environmental endocrine disrupting chemicals in puberty onset. So also reported that the evidence linking endocrine disrupting chemicals is fairly equivocal. There are a lot of limitations due to cross-sectional study design, exposure misclassification, non-standard puberty outcomes, and a lack of focus on chemical mixtures, and called for large longitudinal epi studies. And so this brings us to the study design for MIREC Endo. It's a 3-phase study designed to account for the fact that children go through puberty at different ages. And so we need these 3 phases to capture the fact that puberty timing is variable. Phase 1, which I've shown the ages for there now, is complete, and we're currently doing data analysis. There were 311 children that completed a clinic visit, 280 that completed a questionnaire. Phase 2 is currently underway, and we're in the midst of we'll continue recruitment until the end of March, and phase 3 will start in another year. So at each of these phases, children are asked to participate in a clinic visit, to have their puberty staged, to have anthropometric measures taken, as well as blood pressure, and collect biospecimens. We also ask participants to do a suite of questionnaires on diet, stress, cannabis, vaping, smoking, as well as self-assess their puberty. And we also have collect information on sleep and physical activity. I'll just point out that it's clear that phase one occurred during COVID. And so part of the reason that we have a different participation for questionnaire completion versus clinic completion is that many of the clinics were closed for at least part of phase one. And so we had to pivot to allow for remote participation. And so that's why the self-assessed puberty data is really valuable, as well as the extent of information we can collect from questionnaires alone. As I mentioned, we're also interested in MIREC Endo on the maternal health. And to me, this is a quite exciting piece because most cohort studies that have started in pregnancy follow the children, but very few follow the moms into long-term health. And while we know that pregnancy is a stress test for maternal chronic health, we know quite little about the interplay between the stress test of pregnancy, exposure to environmental chemicals during pregnancy, and long-term cardiometabolic health or timing of menopause. This is data here from a study that looked at hypertensive disorders during pregnancy and cumulative incidence of hypertension in a study with 10 years of follow-up. And what this paper shows is that the risk of hypertension long-term is notably higher in those individuals who had hypertensive disorders of pregnancy compared to those who didn't. As you can see in the table, regardless of your age group, your cumulative incidence of hypertension is at times triple what it would be if you didn't have hypertensive disorders of pregnancy. So it's in this framework that we were motivated to follow up mothers during MIREC Endo in phase one, and we're planning for phase three to do both a clinic visit as well as questionnaire information on the health of the mother, including anthropometry, blood pressure, biological specimens, as well as collecting information on menopause, smoking habits, as well as reproductive history. I'm going to finish up in the next slide or two. I wanted to highlight some of the research questions that we have underway in MIREC Endo, both on the mothers and the children. We have a paper underway right now looking at pregnancy complications and postpartum maternal health, prenatal PFAS concentrations and postpartum metabolic health, as well as prenatal and postpartum metal concentrations in hypertension. And the children will have a number of projects underway once phase three is complete, but right now we're looking at prenatal PFAS in relation to phase one anthropometry and cardiometabolic health, as well as diet as a potential determinant of concurrently measured PFAS concentrations. So in conclusion, these exposures during development and susceptibility may have long-lasting effects, although the effects we're seeing at the individual level may not be large. At the population level, they may have meaningful impacts. I've shared some information on the intersection of environmental chemicals and social and nutritional determinants of health, but this requires continued investigation, and it is a challenge in our MIREC study population, which, as I've mentioned, is a fairly homogeneous group of participants. And MIREC will continue to investigate the long-term effects of environmental chemical exposures on both the health of the mothers and the adolescents as we progress through phase three. I want to conclude by acknowledging all my colleagues and mentors, as well as our funding and the site staff and investigators at each of the MIREC sites, as well as the study participants. And I want to conclude by noting that MIREC is really a collaborative research community. The data and biological specimens are available for researchers outside of Health Canada. And we have researchers at universities across Canada and as well as the US. So if you're interested in doing a research project or learning more about how to go about accessing the data, please feel free to reach out to me. I've put my email there below. And with that, I will stop sharing and I think we